Okay, folks, I know some of you have CSB 332 this week, and some of you have Biochem 311 later this week. And apologies, I've been meeting with some of you, but I've also asked to see others of you, and I'm going to try to get through that this week as well. And the last thing, the second last thing, your group marks are ready, and what I'm going to ask you to do is to come by to pick up your midterm, and I'll be posting your midterm. I'm going to ask that if you get the midterm, the actual test in one answer sheet. So version A, you'll get the answer sheet, short answers, and I ask you not to post it on a third-party website. That's stuff that we want you to see what your answers should look like when you come in to pick it up ahead of time, but we're going to encourage you not to be posting on another website like Oxidia or Student Buddy, etc. The Faculty of Arts and Science actually checks that on a routine basis. They can actually track who you are, and it's something that they take very seriously, and I'm going to ask you not to refrain from doing that. And the reason why we're posting things is for you to get a better idea of how to do better on your next test, not for you to be gaining points with some online service. Okay, so look for that in the next couple of days. I'll be giving you a workshop on Thursday, and the TAs will be marking this as quickly as they can and giving you back your feedback on this as well. We're going to switch gears just a little bit, and this is actually a really interesting series of papers, but the papers I'm going to present to you are actually done in a slightly different way. And I'm going to leave it up to you to judge whether or not you think that the papers that I'm going to be presenting to you and the facts that you're going to have to memorize. If it's been a while since you've learned about prion disorders, some of the things that I'm going to be telling you a little bit about will segue into sort of the next section, which is on fatal familial insomnia. And I'm going to be a little bit of an armchair quarterback. I'm going to tell you some of my opinions on some of the papers that we're going to see, why they were published the way that they were, and we're going to try to understand a little bit about prions. Some of you may have taken either PSL or fourth-year LMP courses, but I want to go back to some of the foundational elements of prions, why we're interested in prions in neurogenomics course, and why prions might be of interest to you. Okay, so I'm going to go through some of the basic concepts of prions. And I have to be honest, when I was in undergrad, I didn't really like my neuroscience courses, and I had to take a fourth-year seminar in physiology. And my seminar in physiology wasn't really much better than my third-year physiology course, and they were kind of like talking about, oh, hello, thank you. That's okay. Well, that's okay. There you go. Thank you so much. And feel free, if there's no line, to just go up and grab some. It'll be pretty informal anyways, and some of this will be a review for some of you. But as a fourth-year student, I didn't really like my physiology class. It was kind of hit and miss. We were learning about hearing, we were learning about auditory processing, and we were learning about vision again, and it was in more detail. And it really made no sense in terms of what I was hoping to learn in physiology. But we had this one guy come in, and he was actually doing research. His name was David Westaway, and he's the only prof I remember, his name. And he was telling us about prions. And it was really, really cool because he was saying, you know, I've actually held a brain in my hands that someone had passed away with prions, and you could actually poke holes in it. It was like that spongy. And it was such an amazing lecture, and it was one of those things that has stuck with me for a very long time. And I think one of the reasons why I stuck with neurodegeneration as part of my research was really because of his lecture on prions, and David Westaway has gone on to become a leader in the field as well. So I'm going to go through some of the theories related to prion disorders, some of the new theories and emerging theories, the classic theory by Stanley Prusner back in 1982. I'm going to revisit Parkinson's disease, and I'll give you some theories related to Parkinson's disease and this one protein that I'm sure you've all heard of several times now, alpha-synuclein, and introduce some other genes to you. Then I'm going to, as we normally have done, give you a paper. And I'm going to leave that paper to you, and please feel free to just grab coffee. Some of you actually look like you really need it. Yes, you guys need it. Go for it. And I'm going to actually present it as a series of papers. Okay, and it's up to you whether or not you think this is a really cool paper or it's not such a cool paper. And then I'll give you a really quick introduction to sleep, and then I'll give you an idea why we're actually doing these sorts of papers. So when I heard about this talk back when I was in fourth year, one of the things that really struck me about this was um, they were trying to prove without any doubt that prions actually were transmissible. You could take it from one person and give it to another person. And this brand new class of prions, or these proteinaceous infectious particles, they were very, very clear that these were not viruses. It wasn't a contamination by a virus. So there was no genetic material. And when I heard that for the very first time, it was like, wow, this is like a really, really cool disorder. And these infectious particles were only made of proteins. And proteins were the only things that were being transmitted from one diseased brain to another. So all of these things, again, fall into a specific category. They're resistant to nucleases. There's no nucleic acid, therefore they are resistant to nuclease attack. And in addition, it didn't make a lot of sense to me at the time, they're also resistant to proteases. So they're made of proteins, but proteases don't work. Nucleases don't work as well. So this specific protein that was involved was this prion protein. I was surprised to learn, or perhaps not surprised to learn, yes?
You said, okay, then we're good. Not the proteases. You were surprised to find out that they were affected by proteases?
So they are resistant to protease degradation. I'll show you a figure that's a standard in practice to demonstrate that they are protease resistant in a few slides.
So that statement isn't, but not to proteases. They are resistant to nuclease attack, but not to proteases.
So I'll show you that, but they are actually more resistant to proteases in a moment. So yes, Anne, you got me again with another typo. Thanks. And I'll make sure to clear that up for you as we go along. So this was the big paper. It came out in 1982. I hadn't read it. I wasn't actually in university. I didn't really care. And it was a science paper. It's considered a classic paper. It was published in Science. And Stanley Prusner was widely ridiculed for his idea that proteins could actually cause disease. You could transmit proteins from one individual to another. And Stanley Prusner's idea was that this disorder, when someone came in complaining that they were losing their mind, they didn't have good cognitive facilities, they were having problems with their autonomic system, they were having issues also associated with their ability to walk and have motor coordination, that these transmissible, because you could track how individuals were getting this, these transmissible spongiform encephalopathies, because the brain at the end literally does look like a sponge, these TSEs were a group of different types of disorders that were caused by proteins being passed along. Sometimes those proteins were ones that were inherited. So we know that there are a few forms of prion disorders that are inherited. You get it because you have inherited a mutation in a prion protein that causes more misfolding, and you get a prion disorder. And that's the topic of the next talk, which is fatal familial insomnia. Others are things like Creutzfeldt-Jakob disease. At the time, I called it Jakob-Creutzfeldt. They switched the names around. And we know that it's all due to a misfolding in these prion proteins that are normally found on your cells. And this is sort of a diagrammatic view that you'll see several times on the normal prion protein. And as it became aberrantly folded, that it became sort of this prion scrapie or resistant form of the protein. There was a lot of skepticism about this. In fact, even though he came out with this dramatic hypothesis in 1982, he wasn't awarded the Nobel Prize until several years after the fact, because a lot of people didn't believe him. They thought his methods were wrong, or that there's something inaccurate or missing about a virus that was also being co-transmitted. You take it for granted that there are such things as prions, but neuroscientists up until the 1990s actually highly doubted that prions actually existed. We do know that prions are part of the normal cell. Now I'm actually highlighting a couple of different proteins for you here. So at the top is a totally different protein. This is the amyloid precursor protein that's often associated with Alzheimer's disease. And many of you who have taken a neuroscience course know the structure, know about the alpha, beta, and gamma secretases that take apart this particular amyloid precursor protein, also found on the cell membrane. Similar to that, though, in structure, is this protein down here. This is the cellular form of prion protein, or PRPC. Every cell that you have, especially those in the brain, have this as part of the membrane. So there's an amino terminal, there are these copper binding domains, which are these repeats that you see here in blue. And then in addition, there is a toxic fragment. And similar to what you see here with amyloid beta, or the amyloid precursor protein, if you were to just isolate these particular fragments of the prion protein, the fragments between amino acids 108 to 126, these are the toxic fragments for prions. Just like you have the toxic fragments of the A beta shown here that is associated with Alzheimer's disease, we have exactly the same mirror or parallel with what's going on in the prion proteins. And again, there are lots of similar structures, similar motifs. If you take a very careful look at what happens in the amyloid precursor protein, there are also copper binding domains. It's one of those things that has been noted for a while. I'll just point this out for you right now. This mutation here, the D17D8N, is the type that we're going to look at. It's a mutation that is inherited in some individuals in the prion protein that causes the protein to fold and become more toxic more quickly in some individuals, that causes fatal familial insomnia. And so this is sort of the structure of the protein, just to give you an idea. It's a normal endogenously occurring protein. It's membrane associated. There is a GPI anchor that connects the protein itself to the cell membrane. And so it's very, very similar in many ways to the amyloid precursor protein. So one of these hypotheses that was developed by Stan Prusner, there's no genetic material. So how does it pass from one brain to the other? One hypothesis that he suggested was that this is the mechanism by which a protein can actually cause other proteins to become similar to it. No requirement for having any kind of genetic material or DNA or RNA like you would have with a virus. You simply have to have a protein, and here is the cellular form on the left. And for those of you who are taking Biochem 311 and you have your term test on Thursday, so what kind of structures dominate here? Or Biochem 210, same thing. So you have about 40% of the structure of that protein are alpha helices, right? And you can see these alpha helices, one here, one here, one here, and one there. A small change, a very, very small change, causes this protein, which is relatively short, it's only about 200 amino acids long, to take on this conformation that you see here on the right. And what do you see here on the right? So you see a lot of beta pleating or beta sheeting. And so one of the things that we know is that this form, the beta sheeted form, the scrapey form, is actually much more stable in solution, and it is something that accumulates over time. This form here on the left tends to be more monomeric. It doesn't like to clump together, but the form on the right is resistant to protease attack. It's also very stable in solution. It doesn't get degraded very easily, and it likes to stack up on top of each other. So these two different types, the cellular normal isoform that you see on the left, every cell has it, including cells in the digestive system. And these are sensitive to being broken down by proteases, etc. And the scrapey isoform that you see here that has so many of these beta sheet structures are more resistant to being broken down by proteases, as well as being resistant to nuclease attack. Is that yes, Jenna? That's a great question. And in fact, we don't know entirely. The beta pleated or the beta sheets themselves don't necessarily confer any advantage to being protected by protease activity. Some of it has been thought that when they clump up together and when you start forming these beta-pleated sheets, it makes it easier to stack, like Lego blocks. And as a result of that, it makes it harder for the protease to actually get in. But this is true for the most ubiquitous protease we use in the lab, which is proteinase K, and I'll show you this diagram in a few moments. And proteinase K actually doesn't care what sequences are around it. It's just a ubiquitous protease, but even that doesn't tend to degrade or break down this particular protein very well. So somehow, and this is the big thing, Stan Prusner was able to show, and before I get to some of the other slides, what prion diseases other than Jakob-Creutzfeldt or Jakob-Creutzfeldt are you aware of? So there are two forms, by the way. There's a variant of Jakob-Creutzfeldt, and then there is sort of the endogenous form. What other one do you know? Kuru, okay. Kuru is another, yes. Mad cow, sure. Let's talk humans, but you're absolutely correct. Should have phrased my question better. Lauren? Okay, Parkinson's, definitely. That's the topic of today's paper. And anything else? Any other prion diseases in humans? Yes, at the back. Fatal familial insomnia, absolutely. Anything else that you guys are aware of? Yeah, also potentially Alzheimer's disease. A lot of, well, maybe not a lot of controversy anymore. Yeah.
Isn't there less common one that's thought to be transmitted from eating deer?
Yes, in fact, this is one of the things. How do you transmit it? So normally, Kuru is something that you get transmitted to you because you've eaten someone else's infected brain. Probably not going to happen, but you can also eat other infected animals, and the species barrier, which is something we're not going to be talking about, doesn't seem to apply in this particular case. Yes? GSS, and we'll get to GSS in a moment. Some of those are genetic. Some of those are not genetic. Some of those are ones that somehow you have ingested. So the spontaneous forms are the ones that are inherited. You inherit something, something happens to the prion protein, it starts to spontaneously form the scrapie version that is known in fatal familial insomnia. GSS is another form that is inherited. And sometimes you get inoculated. You eat something that's contaminated. You eat meat, for example, that is contaminated by mad cow, which is a prion disorder. You eat that and it gets up into your brain, and it also causes the prions normally found into your brain to start being converted as well. So we're not 100% sure of the routes of transmission, because you have to think about this a little bit. Do you know how things get from your stomach up into your brain, right? Because this is the traditional route that you are getting transmitted to you. So you've eaten something that's contaminated. You ate a burger that had meat that had mad cow. 10 years later, you start feeling weird, your cognition goes, you no longer have motor coordination. You think back to that really delicious tasting burger that you got from, I don't know, In-N-Out Burger down in the States when you visited really briefly, and it turns out that you've ingested something and it's gotten into your brain. How does it get into your brain? How does it go from your gut into your brain? You must have thought of that at some point, right? Like, how does it go from eating something to getting up into your brain? It's not like you're not, even if you eat someone's brain, like in Kuru, you still have to get it from your stomach up into your brain. How does it get there, Chelsea?
Well, if it survives its way into the intestine,
Yep.
if it's really small, it could get taken up into the blood by
Yep.
The normal systems that do that in the villi, and then it would just have to pass the blood-brain barrier somehow.
Possibly. Okay. Possibly. It's a good idea. Yep, Richard. It potentially could. There's been no suggestion that it actually gets taken up by the vagus nerve. It's a good idea, and it retrogradely transports. No evidence of that yet. Anything else? Those of you who are taking immunology. So what sorts of things exist in your intestine? So you have intestinal cells. You have immune cells. Do you have any lymph nodes down in your intestines? So we have various lymph nodes. We have Peyer's patches, and it's thought that there are certain cell types, dendritic cells, that are part of your immune system that sit in the lymph nodes. And we now know in 2015 that there is a lymphatic system that exists within your brain as well. And it's been thought that that's the route of entry, that from your stomach, the lymph system actually carries it up through contaminated dendritic cells, etc., that get into the brain. And it's still very, very hypothetical. But again, I want you to think like integrative neurobiologists. Like, how does this go from your stomach up into your brain? I think this part, most of you have a conceptualization that if you have this misfolded protein, it acts as a template and misfolds other proteins that it comes into contact with, which is the protein-only hypothesis that was put forward by Stanley Prusner back in 1982. Sorry, just trying to get the laser pointer to come back. Have you guys heard of the protein-only hypothesis? One protein becomes a template for normally existing proteins, like prions, which then cause aberrant misfolding. So it converts the soluble PRPC into the insoluble or protein-resistant PRP scrapie version. Okay, so it's like a template. As long as you have a small number of these templates, these will convert some of those into the scrapie version. And again, there's a lot of controversy still, many, many years out, as to how this actually works. Does it have to be endocytosed? Does it happen on the cell surface? Does it happen inside the cells after it's taken up? Does it happen in the level of the lymphoid cell, where the prions are found in the lymphoid cells, and then they get converted in the lymphoid cells, which then take it up into the brain, and then it converts that in the brain? We're not really sure about all of these. We're only fairly recently able to say that the lymphoid system seems to be involved, whatever that lymphoid cell is, whether it's a dendritic cell or other type of lymphoid cell. And sometimes there is some idea that maybe these cells burst open, that when they're accumulating them inside, that the prions, after a while, they aggregate inside the cell, and as the cell dies, it starts to burst open, and that's how these aberrantly folded scrapie versions that were found encountered first in the stomach spread throughout the rest of the brain. So I'm going to ask you again to think a little bit about what you know about neuroanatomy when I get to this in a few moments. So these scrapie isoforms, getting back to your question, Jenna, they tend to be a little bit more adherent to each other. So it might just mean less access. And over time, as they start stacking up, these scrapie resistant forms form these longer chains, which in a lot of ways look like those amyloid fibers. When you hear about beta amyloid, and you hear about these beta amyloid plaques, these are very similar looking in terms of these scrapie protease resistant molecules. These amyloid fibers that are formed from prions are believed to be toxic to cells. How they induce cell death is also a source of controversy, whether it's caspase 3, caspase 9 dependent, or whether it is a necrotic factor. It's probably a little bit of both, to be honest. If you take a look at the brain, there's no way that it can only be apoptosis. The amount of cell death is really, really severe. You can literally put your fingers through the holes that exist in the brain of an individual who has advanced spongiform encephalopathy. And part of those holes are because these microglia and resident macrophages are believed to digest as much of the tissue, including some of the extracellular matrix around it as well. So again, it's a really fascinating disorder. I wanted to give you a really quick sort of review, if you haven't had this in a while, on what this all looks like. This is sort of what I've been talking about, and I'll get to this at the bottom in a moment. So how do you actually get prion diseases? Well, one, we're going to be talking about it from a neurogenomics perspective. The second bullet point, which is you can have an inherited mutation. It's in your genome, that over time, some of your normally occurring prion proteins are going to spontaneously form the protease-resistant scrapie form. Very, very rare, only found in 2 disorders that we can identify so far, including things like FFI as well as GSS. And there's something wrong in your inherited prion gene. The other way to have this done is that you eat infected tissue. That's, again, one of the uh, most likely ways to encounter this. If you eat infected cow tissue or deer or something else, then you can infect yourself with these scrapey versions of this, which then cause your own cellular versions to start aberrantly misfolding as well. There have been reports that you have spontaneous mutations developing as well, or the formation spontaneously that is not due to a new mutation, but for whatever reasons in your brain, spontaneous formation of the prion scrapie form starts to appear as well. So if you were to take a sample of tissue, brain homogenate, and you were to run it sort of on a gel, and this is a standard type of gel that you would run for any type of protein, and you were to incubate normal cellular prion proteins, normal form, the things that are normally found in your brain, on your nerve cells, in your lymphoid tissue, and you were to run it on a gel without having any proteinase K, or with proteinase K, you would see that the proteinase K, for the same amount of protein, completely obliterates prions. So they loaded protein in this well, but none of those proteins actually survived. It is labile. It is soluble to this protease. Now, if you take a scrapey version, the one that is aberrantly misfolding, and you run this again, there is this section here, which is the resistant core. It is resistant to protease attack. So you add the proteinase K, which normally wipes out any of the prion proteins. But now when in the scrapey form, if you load exactly the same amount, you'll see that it is much more protease resistant. And we still have a lot of the prion proteins around. And so this is one of the reasons why we say it's nuclease resistant as well as protease resistant. And hopefully that answers your question. Anne, are you good? Thanks. Okay, so in terms of histology, I'm not sure. I don't think they have these anymore in the Grant's Anatomy Museum. For a while, they used to have actual examples of these brains that were available. But if this is a normal brain and this is sort of the normal appearance on an H&E stain, this is an individual who has Kuru or was diagnosed as having Kuru. This is that classic version of Creutzfeldt-Jakob, and this is the brain of a sheep that has scrapey. You can see that there are lots of holes in it. There are lots of areas where the tissue is no longer patent, it's no longer there, and we get this sponge-like appearance on a microscopic and at a gross level when you take a look at it. And again, the Kuru is transmitted through consumption. There are lots of ways you can get Jakob-Creutzfeldt. There are other things like encephalopathies in all sorts of animals. Wherever you look, in deer, in cats, etc., in elk, we can see different types of transmissible encephalopathies occurring. And encephalopathies that have this spongiform appearance are all caused by prions, and they all have this sort of similar appearance. So right now, we don't have a really good way to tackle this. It's just a protein that normally exists in your brain, so you can't really develop antibodies that are specific for this really easily. You actually don't develop an immune response to the pathogen, which also kind of makes it unique. You don't develop any specific antibodies, etc. These spongiform diseases are contagious. They can be transmitted. It's been shown time and time again that if you eat contaminated meat, you will have a prion disorder. And the incubation time, the time that it takes for scrapie to get up into your brain and then cause the normal prions to be converted into the scrapie form, there is a very, very long incubation time. So when you start hearing about contaminated meat and you have heard that in the news, if you stop eating, it may already be too late. You might have to wait 10 years before you have autonomic disorders, problems moving, etc., where it might be related to you developing spongiform encephalopathy because you've eaten something that's contaminated with the scrapie prions. These spongiform diseases are, yes? Yeah.
Is it noticeable when you eat it? I mean,
I don't know. Having never eaten it, I don't know. No, no. Actually, there's no way for you to tell. Nope. 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 There's no. The meat doesn't glow green. There's nothing special about it. It's just like normal meat. Don't know. So this is, yeah, it is scary. It's fatal, and it's also untreatable. As I mentioned, there is no cure for this. There are lots of individuals who were infected during sort of the mad cow outbreak in the UK, and then 10 years, 12 years later, they started developing signs that they had encephalopathies, and we know that it is a fatal disorder. There are no effective treatments. Once you're diagnosed, it's all downhill. It's a progressive disorder. And the symptoms actually are really interesting, right? So prions, you think a little bit about this and think like neurobiologists, think like scientists or researchers. These prions are invading your brain. But we also know that where they are starting to be concentrated make really big differences. So you have a prion, the same sort of prion that gets aberrantly misfolded in fatal familial insomnia. And if you were to take out proteins from that person's brain and put them in someone else's brain, they'll also have scrapey isoforms developing. But they actually have a very unique disorder where they start to not be able to sleep. So that means that there's a specific area of the brain where these prions are being built up, causing losses of neurons. So think a little bit about the neurobiology that all of you have as third-year students. Where do you think prions are more likely to cause damage in something like fatal familial insomnia? They don't really have like motor coordination issues, right? It's not affecting their cerebellum like GSS or other disorders. Here it's affecting what area of the brain? Yep. Could be the hypothalamus. Yep, definitely a target. Why do you say hypothalamus? Sleep areas in the hypothalamus, sure. And we'll talk about that in the next lecture. Eddie? Also the thalamus. Yep. Lauren, did you have a different answer? Okay, so we're all talking about the area around the thalamus, and it turns out that that indeed is the region where we get in fatal familial insomnia, the groups of neurons are affected there. We see the highest concentration of aberrantly folded prion proteins there. So the symptoms, even though prions are misfolded proteins, Eddie, do you have a question? Yes, actually it does. It turns out that it does, but we'll talk about that I think today, and then I'll bring it up in the next lecture as well. But it is important where you actually incubate this. Okay, so preventing it, just don't eat any of the infected tissue as long as you know that it might be contaminated. And again, there are some signs for mad cow disease. We often get it in Canada. Out in Alberta, you'll often hear warnings about mad cow. Then they take all the beef off the market for a while until they don't see any neurological signs of any infected tissue. It turns out in the past that sterile surgical instruments have been infected as well by prions. And prions are really kind of neat in the sense that we can't get rid of them. They're very, very resistant to traditional boiling. So if you're sterilizing through boiling only, it doesn't work. Alcohol doesn't seem to work. Acids, they're protease resistant, also acid resistant. Standard autoclaving, where you're only taking it up to, say, 110 degrees, not enough. You have to take it all the way up to about 120 degrees before you can actually destroy prions. And even radiation hasn't been shown to be that effective. So how do you make sure that meat is sterile? You irradiate it, right? You get that gamma irradiated meat, or you cook meat, etc., and you're supposed to sterilize it that way. Doesn't work. Doesn't work at all. And it's been thought, and it's been reported at by the University of Utah, that this is a stable protein that brains that have been sitting in formaldehyde for decades, and you can take tissue from that brain and then still transmit the disorder to a living brain. And again, think about how stable this protein is. If only the molecules that were involved in learning and memory were that stable, right? If you were able to transmit learning and memory molecules that way, that'd be awesome. But this is a disorder. It's a disease that has no sort of likelihood of ever being cured if you are diagnosed with it. So one of the other things that makes these disorders interesting for us today and then into next week, we see amyloid deposition. In fact, amyloid deposition, when you hear about amyloid, these are just proteinaceous fibers that are found outside cells. That's really all amyloid is. Sometimes those proteinaceous fibers contain beta amyloid, and that's still an amyloid fiber. Sometimes these things contain prions. It's still an amyloid fiber that's found outside the cell. And these amyloid deposits, even before we had modern immunological techniques, we knew that in brains of individuals that had neurological issues, we would be able to detect amyloid particles. Do you guys remember what the histological stain that you can do? You can just slice tissue and you can stain it. You don't need antibodies or anything else. And it's an old histological technique. Do you guys remember that at all from any of your lectures? Sorry? H and E. No, not in this case. Doesn't work. Yes. You could do silver staining in some instances. It won't work for all amyloid deposition, but do you guys remember Congo red birefringence? It's like an old technique, but it works. I mean, if you have amyloid deposition and you stain with Congo red, it's a red stain as the name suggests. You dip your slides in it, then you put it under a microscope, and then you hit it with UV light. It fluoresces if there's any amyloid. And we know that diseases, even before the advent of antibodies, diseases like Alzheimer's disease had amyloid deposition that we could see through Congo red birefringence, that you could see things like amyloid in Creutzfeldt-Jakob disease. We could see this in Down syndrome. We see lots of amyloid deposition in Down syndrome. We know that in FFI, or fatal familial insomnia, or GSS, Gershman-Straussler Schenker syndrome, as well as Kuru, and even in leprosy, we can see amyloid deposition in the brain of these individuals. However, if you take a look, a lot of these actually involve these prion diseases. So Jakob Creutzfeldt is one that is transmissible but is not genetic. FFI is genetic, and GSS is genetic in origin. And we'll only be talking about fatal familial insomnia. But all of them involve these changes in prions. Some of you had mentioned when I asked you about some of these other types of molecules. This paper I'm going to show you, actually there are two papers. They're weird papers. They may not look weird to you, but when you're sitting back and you're kind of looking at these papers, they are weird papers. And I actually pulled them for reasons. Papers, Stanley Prusner, who was the first person to come up with this protein-only hypothesis back in 1982, actually is the same author that's on the two papers in 2015 that I'll be going through with you. It kind of sounds like he came up with this idea that neurodegenerative diseases are also caused by prion-like proteins. Here, the prion-like proteins might be things that are normally associated with Alzheimer's disease. It could be like beta amyloid that is like prion-like, or it might be alpha-synuclein in Parkinson's disease that might be prion-like. An aberrantly folded alpha-synuclein gets transmitted to someone, they develop Parkinson's disease. And it may sound like he came up with that idea, but that theory is actually as old as 1992 and again recapitulated in 2002. And I'll get to that a little bit later on. There is no evidence, though, there was no evidence until 2015 that this theory that, you know, what you're seeing in Alzheimer's disease is actually a transmissible disease. If you have aberrantly folded beta amyloid, it causes other normal beta amyloid to start aberrantly folding, or if you have Parkinson's disease, you see the same types of things going on. But there is no evidence, or hasn't been any evidence, that there is any infectious etiology. There's been no study where you can actually prove definitively in humans that Alzheimer's disease or Parkinson's disease might also be transmitted just like we would with Parkinson's. Because we know in Alzheimer's and in Parkinson's and in any neurodegenerative condition you look at, there's always one misfolded protein wherever you're looking. Copper zinc superoxide dismutase, beta amyloid for Alzheimer's disease, or hyperphosphorylated tau. There's some core protein that is being affected, or alpha-synuclein, as shown down here in Parkinson's disease, might be the prion, or the prion-like transmissible particle. So you would have to apply, if you're going to show and demonstrate that you think, your theory, that alpha-synuclein is a prion-like protein, that something gets aberrantly misfolded in Parkinson's, and then you would have to be able to demonstrate that when you take this aberrantly folded or misfolded protein and you put it into a fresh brain, that it also causes aberrantly misfolded proteins to occur in that brain, just like with prions. However, those types of experiments have not been that successful. There have been attempts to show this in animal models, and some of these animal models haven't really been as successful in terms of being able to show that this candidate protein works just like prions. So of all of those new candidates for these prions, is amyloid like a prion? If I inject you with like a misfolded amyloid, will you eventually, even if you have no genetic predisposition, even if you were never going to get Alzheimer's, would you develop Alzheimer's disease? Same thing if I injected you with misfolded or hyperphosphorylated tau. Are these the types of things that will cause misfolded proteins, extensive amyloid fiber deposition, etc., if I have injected you with this? Can I transmit this to you, in other words? However, whenever we've looked in all of the different studies that have been published to date, and there were a number, there were lots of studies that came out in 2012, 2013, 2014 to try to do exactly this. We take beta amyloid from this animal brain and we inject it into another animal's brain. Doesn't work. Doesn't work in the same way that prions do. They are a millionfold less pathogenic. You'd have to put in like a huge amount of protein, in other words, not just a small amount, like at the attomole or femtomole level. Like we're talking micromolar and higher levels, which is a lot in the brain. To be as pathogenic as scrapie. So that's not so good. It means that it's really not that transmissible. This model was falling apart in everyone's hands. Everyone started to abandon this model that was originally brought up in 1992 and then recapitulated in 2002. It sounded really good for a while, but all the experimental data actually showed, nope, doesn't work. You need too much protein for you to be able to convert a normal brain into a pathogenic brain. Until this series of papers came out. Now, what do you notice about these papers? Anything? Yes? Yeah, so that's the first thing that's weird. Imagine you're doing all the bench work, you're doing all the experiments, and you are Amanda Verman, and you've done everything. And your supervisor comes up and says, you know what, I'm going to be first author on the paper. It's not usually seen where you get someone of Stanley Prusner's Nobel Prize laureate actually putting himself as first author on these types of papers. Can you think of a reason why he might have done that? Yeah?
In case the idea went really, really bad.
The idea went really, really bad? Okay. To cover her. Okay, so he's being like really, really nice. Yeah, he's being noble. So just in case everything just falls apart, I'll take the blame. I'll be first author. Usually doesn't work that way, but good thought. Any other ideas? Yeah? Yeah, so he wants name recognition. And again, at his stage as a Nobel Prize laureate, as someone who's working at the Neurodegenerative Institute down at UCSF, there's no reason for him to do that. Anything else that you notice about any of these other names on here for those of you who are in the neuroscience community? Do you recognize any of the other names here from Toronto? You might have had him as a lecturer. He's here now. So he did these experiments, and these are almost back-to-back papers in PNAS, two different types of things, prions with alpha-synucleopathies and alpha-synuclein prions, almost back-to-back, almost showing the same thing, alpha-synuclein prions, Parkinson's disease. Yes. Joel Watts, yeah. So Joel Watts actually worked with Stanley Prisner for a while. He's now here. I think he lectures in one of the PSL courses or in the LMP course, and he actually talks a little bit about some of his research models as well. So if you have the opportunity in your 4th year course, you should definitely go check out Joel Watts, a great individual, really bright young guy. But you can see that Stanley Prisner came out in a tour de force, back-to-back papers saying almost exactly the same thing, that if I have a misfolded protein that has been associated with Parkinson's disease, this alpha-synuclein, if I transfer this to a normal cell, it becomes like a prion. And I can demonstrate that it can be passed along just like prions are. So this is sort of the unusual but cool papers. The other thing I'm going to draw your attention to, these came out in a very, very short span of time, July of 2015, July 8th of 2015, and then 2 weeks later, July 22nd of 2015. Just keep the 2015 in mind as to why he might have tried to publish this so incredibly quickly. So I know some of you have had the alpha-synuclein lectures with me before, or it might have been a while since you had alpha-synuclein. So I wanted to go back and revisit alpha-synuclein and talk to you a little bit about this. When we talk about prions, we talk about scrapey versions of prions. When we look at Alzheimer's disease, all of you know about beta amyloid. When we talk about Parkinson's brains, we talk about alpha-synuclein. And alpha-synuclein is deposited in and around neurons as well as glial cells, and it's the neuropathological feature that is most commonly associated with pathology associated with things like Parkinson's disease or dementia associated with Lewy bodies. You can also have a condition where you have pure autonomic failure, and oftentimes in these conditions, they all kind of overlap a little bit. And the thing that I'm going to talk to you a little bit about is this multiple system atrophy. Have any of you heard of MSA before? I know some of you have heard of Parkinson's disease, MSA, multiple system atrophy. Okay, so we'll talk a little bit about that. It's all kind of related. They all kind of look like Parkinson's disease. There are problems with the autonomic nervous system. There's problems with movement, etc. And we get the deposition of this alpha-synuclein. So can we take aberrantly folded alpha-synuclein and can we use it like a prion to generate these types of disorders? Or can we use whatever is wrong in these disorders, whatever the target protein is, alpha-synuclein, and create exactly the same disorder? So multiple system atrophy, for those of you who haven't had this before, actually started off as 3 separate diseases or 3 separate syndromes. They all kind of look a little bit like Parkinson's disease. It was a neurological and histopathological separation of all of these. So Milton Shai as well as Glenn Drager had 2 patients in the 1960s that were suffering from orthostatic hypotension. And what is that? It's also found in Parkinson's disease, yes. Yep, you just can't maintain your blood pressure, and it's when you're going from a lying down to standing position, you have hypotension, low blood pressure. That's what sort of system is affected in that? What normally controls like your blood pressure? A lot of factors. Okay, so nebulous together known as hypothalamus. Okay, so the hypothalamus is the master regulator of what sort of branch of the nervous system? So the autonomic nervous system is often affected, and there are other forms of autonomic nervous system insufficiency that's found not only in this, but also in Parkinson's disease as well. And it is also a movement disorder that looks a lot like Parkinson's. It is not the same disorder. It is not only the substantia nigra that gets affected, many other regions in the brain also get affected. In a few years later, almost a decade later, Graham and Oppenheimer suggested that this particular syndrome that they originally described should actually be combined with loss that is found in the striatal nigral area as part of like the substantia nigra, as well as loss in the cerebellum. And one of the things that they suggested was, you know, they all kind of look the same. Maybe, in fact, you're just looking at one piece, I'm looking at another piece. In fact, all three of these things are really the same disorder, and that these three entities should together be called multiple system atrophy, where all three systems, the striatonigral, nigral, the cerebellar atrophy, the things that result from autonomic insufficiency, all of these things should be clumped together and called MSA. And that's how we have looked at it since then. And if you take a look at individuals that have MSA, they all have these very, very characteristic silver positive accumulations, which are known as GCIs. And these glial cytoplasmic inclusions, or GCIs, are found in glial cells, primarily oligodendrocytes. You can also find them in some other astroglial cells as well. But this is the characteristic defining feature of this. And this, again, is the 1990s, before we knew a lot about the different molecules that are involved. So these GCIs, people were describing them, they were doing silver staining, seeing these GCIs, but it was later found that these GCIs were positive for that misfolded protein, this alpha-synuclein. And the discovery that MSA is actually due to an aberrantly folded or misfolded alpha-synuclein protein was actually really, really important news. It suggested that if you have a misfolded protein, whether it's in Parkinson's or whether it's in another neurodegenerative disorder, that this might be the underlying cause for all of these things. So again, other investigations have actually shown that if you have a point mutation in alpha-synuclein at position 53 of the full-length protein, that this can also cause alpha-synuclein to misfold more quickly or more rapidly. And this is also found in inherited forms of Parkinson's disease as well. So I think now is actually a pretty good time to take a break, and then I'll take you through some of the neurogenetics of alpha-synuclein. I'll leave it to you whether or not you think that Stanley Prusner did a good job of trying to show that the misfolded alpha-synuclein actually works the way that he said, and why he actually published the way that he did. Okay, if you don't mind, I'd like to get going, and we'll try to get through some of these different slides. I know some of you have seen this slide before when I talked to you about Parkinson's disease in a different course. I'm going to ask you to do the same thing that I did for the other course in terms of memorizing this slide, which is really to concentrate on things that I'm going to be talking about, which is really this alpha-synuclein. And unlike how I approach it in sort of the neurobiology course, I'm going to look at it from more of a structural standpoint in this course. In terms of its pattern of inheritance for forms that are familial, if you're taking a look, this is normally inherited as sort of an autosomal dominant form of inheritance. We know that individuals who have a genetic predisposition to inheriting and showing a familial pattern of inheritance for any neurodegenerative condition always exhibit this earlier as well as much more severely than someone who has a sporadic mutation. So someone who has familial Parkinson's disease, it's always worse than someone who has it as a sporadic form of Parkinson's. Alzheimer's disease, if you get it as a familial form, always worse than if you have a sporadic form. And so this alpha-synuclein in the familial forms that have autosomal dominant inheritance patterns tends to be very, very severe. Now, when we take a look at that chromosome, and we take a look at it in a little bit more detail, chromosome number 4. This is sort of the mRNA that comes out of this. And the most important region that I wanted to highlight for you, so there are lots of regions that we have mapped out because this is a really important protein. We know that in Parkinson's disease and in multiple system atrophy, that this is sort of the protein that gets affected. And because it's relatively small at about 140-ish amino acids, that it's really easy to map out the functional domains of this particular protein. When we go back and we examine it, these regions here are the ones that tend to be involved in aggregation. So when they start clumping, which is very common in neurodegenerative conditions, it's the NAC domain that tends to cause this clumping. Now, strangely, I just told you that the inherited forms often lead to having mutations in the A53 to T variant. And this particular variant here is in the amphipathic region, where it might be involved to other molecules. And this has been known for a while, or suggested for a while, that that somehow affects the overall aggregation as well as clumping with other types of proteins through its amphipathic region. So we know a lot about the molecule. Overall, it's really small. This does tend to accumulate outside of the cell in Parkinson's as well as in multiple system atrophy. And I wanted you to concentrate on knowing this, not sort of the number of exons, etc., but to more concentrate on what we know about the regions that might be involved in alpha-synuclein. Okay, so that gene SNCA encodes for that alpha-synuclein. It's that first gene that was ever linked to familial Parkinson's disease. Again, Parkinson's disease, multiple system atrophy, many of the areas of the brain are commonly affected. It's the principal protein that's associated with Lewy bodies, which again is a characteristic feature of Parkinson's disease. It's also alpha-synuclein that is found in those GCIs that are found in multiple system atrophy. It's believed to be involved in the pathogenesis in Parkinson's disease as well, specifically in the substantia nigra. And we also know that if you overexpress the gene, the alpha-synuclein gene as well, that it can also cause familial Parkinson's disease. Do you guys remember what that was called? Do you remember the induced pluripotential cells? You overexpress copies of the SNCA gene. You have 4 copies or 3 copies. Do you remember what that's called at all? That kindred? The Iowa kindred? Who said that? Nice, good memory. Yes, so we know that overexpressing the gene itself is enough to cause overaccumulation, which can lead to issues as well. So these genome-wide association studies implicated that this SNCA locus is the one that is the highest risk factor for sporadic forms, not the familial inherited form that I just showed you previously, but also associated with the sporadic form. So there's a lot sort of clustered around this alpha-synuclein. If you do a meta-analysis of different GWAS studies, then you can also see that something that turns on the expression of the alpha-synuclein gene can also enhance or speed up the Parkinson's disease as well. So it suggests that maybe a promoter variation or enhancer region might be affected in different forms of sporadic Parkinson's disease. And in patients with Parkinson's, oftentimes the familial inherited forms of this have a change at position 53, and that leads to that autosomal dominance. So now I'm going to take you back to this paper. So that's alpha-synuclein. We know it's in Parkinson's. We know it's in multiple system atrophy. We know there is an overabundance and accumulation, too much of it outside the cell, similar to what we see with beta amyloid, similar to misfolding that we see with prions. So does this actually work like a prion? Can I take alpha-synuclein from a brain that has MSA, and can I cause this alpha-synuclein to start aggregating inappropriately, just like we see in MSA? If it is, if we can show that, then it's an indicator that maybe it's a lot like the prion disorders that we've just taken a look at. So multiple system atrophy is sporadic, tends to be an adult onset. It's progressive, so once you're diagnosed with it, there's no coming back. It's neurodegenerative, movement disorder similar to Parkinson's. It's not as common as Parkinson's, so it's about 3 out of 100,000 individuals, typically found in individuals over the age of 50. The duration of MSA, once you're diagnosed to the time that you die, is only about 5 to 10 years, so it's a rapid, very progressive disease. It's all downhill, in other words. and it's substantially shorter than in cases of Parkinson's. So if you're diagnosed today with Parkinson's disease at age 50, in theory, you could survive out to your 60s and maybe even out to your 70s. If you're diagnosed today at the age of 50 with MSA, you're more likely to make it to 55 or 60. So it's a much more progressive, rapid onset, once you have it, type of disorder. Not to get into too much with this, more in terms of if you happen to run into Joel Watts in physiology down the road, you can also subdivide this, because multiple system atrophy, because of the way that it was originally described, have ones that seem to be much more Parkinsonian-like, and it's called multiple system atrophy Parkinsonian, or ones that are much more associated with cerebellar dysfunction. So they're targeting different areas of the brain, and different areas of the brain tend to be affected. If any of you are wondering, what did you guys eat from the bakery today? I don't know, it was bread. It was kind of good, kind of tasted like orange, cranberries, chocolate. Not sure. Did it taste like orange? No, not at all? Okay. If you take a look here, in Parkinson's, it's actually really hard to see the loss of the substantia nigra until it becomes pathological. And in an MRI, you can't really see the degeneration of the substantia nigra. On these different MRIs that you see here, you can actually diagnose if an individual has MSA. So there are some drawbacks to having MSA. You have very characteristic progressive loss, and it's very, very rapid, and it's about 5 to 10 years. However, with MSA, not only do you have those gait abnormalities that in many ways look very similar to Parkinson's disease. We know in Parkinson's disease, as the disease progresses, there's also autonomic involvement. But if you put them in an MRI, you can actually start to see the appearance of this hyperintensive rim shown here, this whitish area that you see just to the left of this series of arrows and just to the right of this series of arrows that are found in the dorsolateral putamen, so not in the same areas that you would necessarily associate with Parkinson's. And then a very, very characteristic defining feature in the pons known as a cruciform pontine hyperintensity that you can see very clearly here, or maybe more clearly on your diagram. So you see the little hot cross bun shape in there? And that is exactly the types of things that you see in your pons if you have this MSA. Can you think of why you might see these hyperintensities? So, you know, why does this hot crust bun, and it's called a hot crust bun appearance, by the way, why does it actually appear? What's going on in their brains? What are you picking up microscopically? GCIs, and so the oligodendrocytes are picking up and are being more intense for staining with things like the alpha-synuclein, which is what is thought to cause this sort of unique pattern as someone who has MSA. So there is sort of a method to my madness in terms of what we sometimes eat here in class. So in a previous study, and this is actually where Joel Watts' first paper, he's the first author on the 2013 study that was published. Two individuals that had MSA, diagnosed with MSA, their brains were taken out, their brains were homogenized and then injected into mice, these TG, transgenic M83 heterozygous mice. And what resulted was that after about 120 days, they had progressive CNS dysfunction. So 120 days, it's not bad. And these TG M83 mice are homozygous in this case for a mutation in alpha-synuclein. So we know that there's a problem with alpha-synuclein, and now we have a mutant mouse that we have overexpressing the wrong type of alpha-synuclein. They don't usually do very well, and so we put them into heterozygous mice. And the brains of these mice also exhibited, when they took these out after injecting them, had extensive phosphorylated alpha-synuclein, and it was found in the cytoplasm, axons, etc. It looked like it was accumulating. In other words, it looked like what we might expect to see in the brains of individuals that had MSA. So you were able to transmit MSA from one brain into a mouse model brain. And this was back in 2013. So you might wonder, yes? Yes, 2 copies of the alpha-synuclein that has a mutation that causes it to more likely associate or clump. Okay. So there are a couple of reasons why they decided to use this transgenic. So this transgenic mouse that has a mutant alpha-synuclein, which is normally a problem. If you had it, you would likely develop Parkinson's disease by the time you reach 50 years of age. These transgenic mice do not spontaneously develop Parkinson's, which is one of the reasons why they use this. Yes?
In the previous slide, I guess in this slide as well, isn't the concept taking the mutated alpha-synuclein and then putting it into a relatively healthy mouse and then watching if it can develop that way? I think I'm getting the whole concept wrong.
So what you're really doing is this. So you have a mouse, but it's actually containing a human alpha-synuclein, right? And in this case, you're putting a human alpha-synuclein from someone who has a disease, MSA, into the background of alpha-synuclein from a human source. So they're transgenic animals that have a human copy.
But the human copy is already mutated.
Correct. In this case, it is a heterozygote, so they're carrying half the gene load.
Yeah, in the previous one where there was like homozygous but mutation.
So in both cases, though, no. So I'm just mentioning what the mouse actually is, and these mice don't do very well. They don't survive very well. They have full-blown Parkinson's. But if they have full-blown Parkinson's anyway, and you want to see if they're developing something that looks like a gait disturbance, you wouldn't want to use them, right? And so you use these heterozygous mice that don't normally have that, and then you inject in. Do they actually develop disturbances? I'm hoping that makes sense for everyone. So one of the things that they decided to do, they inoculated these mice with all of these other patients that they had, not just 2, but a variety. And again, you might think, is it really all that novel or really all that? The majority of the patients that they took the brain samples from, the MSA patients, some of them had Parkinson's-like syndromes rather than the cerebellar-like. Remember I told you the 2 classifications, the MSA-P type and the MSA-C type. There's actually a difference. And one of the reasons why they report things like this, so in the UK, Australia, and in the United States, Western countries, it is more common for you to have the Parkinsonian type. And in East Asian countries, it's more likely that the clinical presentation will be of the cerebellar type. Okay, so this is the reason why they're showing you all of these different things and why they had Parkinson's-like rather than cerebellar-like symptoms in the clinical presentation. So again, if you're taking a look, this is the substantia nigra shown here on the right, and you can see the diagonal band of neuromelanin containing dopaminergic neurons that are part of the substantia nigra. And you can see that even in MSA, you're still losing dopaminergic neurons. You're still losing. You don't see that dark band. The dark band is disappearing. These neurons here, you're losing them because they're being lost through neurodegenerative processes, probably because the cells are accumulating alpha-synuclein. If you go and do histology on an individual who has Parkinson's disease or MSA, and this is patient number 10, Parkinson's disease patient number 6, MSA, you can see that overall the brain histopathology, even without looking at it in a lot of detail, you can see that the Lewy body inclusions that you see in Parkinson's disease on the left panels are very different looking than you see in MSA. And when you start looking at the brains of individuals with MSA, they all exhibit that phosphorylated alpha-synuclein, and they also see that they are accumulating in the brains of these individuals. So all of these individuals, in other words, had aberrant alpha-synuclein. Okay, and that's really all you should be taking away from this. So these are the brain samples that they want to use. Can we transmit this really dysmorphic alpha-synuclein? Can we transmit this now into the brain of our mouse model? So again, without going into a lot of details, if you take a look at these frozen brain samples, and you can take a look at Parkinson's disease patients over here on the left, and you can take a look at patients that have MSA. You can see that they all sort of have insoluble aggregated phosphorylated alpha-synuclein, and that's shown in these middle panels. So a couple of things that you should be mentioning or thinking about. So in Parkinson's disease, do you see phosphorylated alpha-synuclein? So these bands here, do you see these bands there? Not really. You don't see.
Well, yes, but I mean, how much in comparison to the other bands?
So if you take a look at Parkinson's disease patients, you do definitely see phosphorylated alpha-synuclein aggregates. So these are bands of phosphorylated alpha-synucleins, or these, or these. So they do exist in Parkinson's disease. In an MSA, you can see that depending on whether you're looking at patient 3 and the basal ganglia or substantia nigra, that you might have more or less. But they definitely have alpha-synuclein. And these are the bands in the middle that you're looking for alpha-synuclein. So they definitely have it. They then will take these homogenates from these patients that have a lot of this alpha-synuclein, even though they're variable. So there are darker bands here, there are almost non-existent bands here, there are darker bands here, and in the middle for this individual, they have almost none. And they will then take the insoluble fraction and then start to manipulate those. So again, before they started this, they wanted to know in those individuals outside of the alpha-synuclein that they were able to characterize that they expected, did they see anything else going on in their genome? So they definitely expected to see some sort of accumulation. They did through biochemistry. They didn't see any duplications, triplications, or missense mutations, and that's that Iowa kindred where you overexpress it. They didn't really see any of these single nucleotide polymorphisms that have been identified, nor did they identify through genome screening any mutations in coenzyme Q10, which is also seen. So they wanted to make sure if it's going to be transmitted, it's only because of alpha-synuclein. It's not due to anything else going on like coenzyme Q. This is the only reason why they're developing problems in their brain. Okay, so they have a couple of different assays. This is their standard assay, and if you go to any of Joel Watts's talk, he still uses this assay in sort of a background screen. So they have these HEK cells, or human embryonic kidney cells, and these are each one of these little blobs that you see here in green is a human embryonic kidney cell, and these have a alpha-synuclein where they have YFP, which is why these cells are fluorescent green, which also contain, just like the mouse, the alpha-synuclein that is more likely to clump. Do you see clumping in the control? Clumping? Or is it just like a uniform green that you see everywhere? Uniform green everywhere, so all the cells are uniform for the alpha-synuclein. And if you were then to take and apply some of the brain homogenate directly onto the cells, and you wait long enough and you give them the incubation time of like 110, 120 days, one of the things that you should expect to see is the alpha-synuclein starts to clump if indeed it is transmissible. And when they start to do this, they're actually able to show that the alpha-synuclein that normally should be nice and uniform actually starts to aggregate. You see little splotches, as you see here in MSA10, of these little bright fluorescent regions. So this is a good indication that maybe this is actually working the way that it should. And forget the technical details about using phosphotungistic acid to cause precipitation. It's just one way to purify your sample. Do you have any questions on this at all? Yes, Anne.
So the 6 panels that are labeled with your NSA, so
Yep.
they basically just took the plant and threw it onto the cells. That's what they saw.
It's pure protein. Yeah, it's not plasma. Or did you say plasma? I heard plasma. Oh, it's not prions either, but it's their prion-like protein, the alpha-synuclein. They're trying to show that it's like a prion, the alpha-synuclein is transmissible and causing misfolding. And you can see anytime you see brighter fluorescence, it's an evidence that clumping is occurring potentially like a prion. Do you have a question, Lauren?
If you were to transplant dopaminergic neurons into a brain
Yeah.
of, like, let's say, for example, Parkinsonian mouse,
Yeah.
and then those neurons sort of become infected. Would that also prove?
So to be honest, no one used to think about those things. So if you take a nice healthy neuron and you're putting it into the background of a disease, all you were looking at in a long time ago was, let's just make the disease better. There was no thought that maybe something in the environment could infect those nice healthy neurons. If these papers are right, those things have to be taken into consideration. You're going to have to not just take healthy neurons, but perhaps create a transgene that you're also delivering with your healthy neurons. Don't know. I mean, those are valid questions, and questions that up until a few years ago we never thought of. It's just like, oh, stem cells? You're lacking dopaminergic neurons? Let's put in dopaminergic neurons and recover things. Let's not worry about whether or not the disease neurons that are there might infect the ones that you're transplanting in. It's a good question. Yep.
And on that, did anyone think about doing the reverse in the sense of putting in healthy alpha, so like into the brain with something that already has an infected alpha-synuclein? Like right now we're taking healthy and injecting messed up and watching mess up everything. But if we inject healthy into mess up, can the healthy act like a prion and correct the mess up? I don't know how.
Yeah, it's a good question. A lot of it goes into bioenergetics and how favorable one molecule is over the other. If you have a certain threshold amount of sort of a prion, like a scrapey prion, it will cause, no matter how much you throw at it, it will cause all of those to eventually be. So we're not sure. If this model does follow the prions, it would actually suggest that the healthy ones would eventually become, let's not use messed up, but also aberrant. Okay. Okay. So again, all of these MSA only homogeneous, not the Parkinson's disease homogeneous or control homogeneous, also showed depositions similar to the ones that you see here for phosphorylated alpha-synuclein. In mice, particularly within the brainstem when they go back and do antibody staining. And again, we won't worry about the astrogliosis, which is very, very common. When cells die and when cells are injured, we typically see a glial response. We see an increase in GFAP expression, and that's just a normal sort of histopathology that goes along with it. So they're able to show in an in vivo model when they put in MSA and not a related Parkinsonian disease variant. When they put this in, the alpha-synuclein actually causes accumulation of alpha-synuclein that also gets phosphorylated, just like we would expect for the disease situation that resembles MSA. But really, this is great and it looks great on paper. But really, you have to do the experiment that I told you at the very beginning, that that Stanley Prusner did at the very beginning. You have to do what is known as serial propagation, and it is the defining feature of authentic prions. So it's great. You have just taken human brain tissue that is diseased, MSA containing alpha-synuclein, aberrant alpha-synuclein, put it into the brains of mice. You've just shown that their brains also get messed up. They overexpress or have aberrant phosphorylated versions of this, but that's really not enough. To show that it is transmissible, you actually have to do it one step further, and this is really the characteristic feature which was not done in the original 2013 paper by Joel Watts. And one of the reasons why they had to do this experiment, once you have infected mice and you've shown that you've infected these mice, if it's really that transmissible, those newly infected mice, if you take their brains and you can show that those infected brains can also infect, that is really the sign of what a prion is supposed to be. So this second passage, when they take out the brains of these infected mice, which should contain aberrant accumulations and phosphorylations of alpha-synuclein, when they did this, they found that these MSA-like prion molecules were just as effective. So remember that earlier statistic that I threw at you, that when people did this originally, that it was like a millionfold bigger than they had expected. But what they found was that when they looked at the level of the aberrant accumulation of these MSA alpha-synuclein prion-like molecules, that they actually saw 1,000-fold amplification. So that over 2 passages from mouse 1 then into mouse 2, the prions had actually multiplied by 1,000,000-fold, which is exactly what you would expect. You don't want to have like 1,000,000 times to infect. You want to actually show that one infection has increased 1,000,000-fold, which is exactly what they're able to show. That from one mouse to another, it's becoming more and more infectious. The number of particles seems to be. So it was a great paper. I don't know if you think it's a great paper or not. Thumbs up, thumbs down. Really novel, really new. Really new. Pretty cool. Can I get a thumbs up or a thumbs down before you? It's a good paper? Yeah. Everyone looks like they're kind of using lighters. Do you have a question, Ann?
So in the first bullet,
Yep.
so the heterozygous mice had inoculation from MSA or the homozygous brains. Is that what you mean?
Yes.
Okay.
So these mice become spontaneously sick. They get Parkinson's-like syndromes. So if you take their brains and you grind them up and you transmit it, do those brains become sick? No, they don't. It's only the ones that you've infected. So any old sick brain doesn't work. It has to be the one that you've infected. And that's the point of that first bullet point. Eddie, do you have a quick question? So these get sick because you've infected them. The other mice spontaneously get sick. The homozygous ones get spontaneously sick because they have like 2 copies of the nuclein that will cause Parkinson's-like syndrome. So they get sick. They develop Parkinson's-like syndromes as they get older. You take their brains, and can you transmit them? You cannot. Okay. It's only the ones that you've infected with the brains from human MSA patients. Does that make sense? Hemizygous, they normally shouldn't get sick. They shouldn't develop Parkinson's. You are making them sick because you're injecting in the MSA from infected individuals.
So what is the distinction between the heterozygous mice that have the mutated alpha they brought in versus the ones that
That's a great question. That's a great question, Danny. And in fact, I'm pretty sure they have a follow-up paper to this, but I don't know the answer to that. Okay, so a lot of you kind of put up your hands that it was a really good thing. It's not. It's a pretty good paper. 2 back-to-back PNAS papers is pretty good. But in fact, this is the better paper. This paper, which came out because they knew that this paper was going to come out. Stan Prusner knew ahead of time that late in September of 2015, or the middle of September of 2015, that another group, John Colling, is actually a really big name in prion diseases now as well, just as famous as Stan Prusner, is going to come out with this paper. And actually what they showed was a very, very different sort of thing, okay? I don't know if you believe Stan Prusner's transmissibility, but what John Colling's group actually did was show everything that Stan Prusner should have shown. And what they showed was actually kind of cool. I don't know if you read this paper or not. Did any of you happen to read this paper? It was like really, really big news. It was all over the news circuit. It suggests that you can infect someone who has Alzheimer's disease by giving them tissue that came from an individual with Alzheimer's disease. Not like what Stan Prusner. So Stan Prusner hasn't actually shown that Parkinsonian-like syndromes can be transmitted in humans. What they were able to show is that a long time ago, for individuals that had, you were short in stature, you had to actually remove the pituitary gland from cadavers, extract human growth hormone before recombinant human growth hormones became really popular, and then inject them, and it allowed for them to have normal stature. Unfortunately, in the quality control process, an individual was identified who had a form of a neurodegenerative condition. His pituitary gland was also, because it contains neurons, his pituitary also was contaminated, and those individuals that received injections from his human growth hormone actually developed Alzheimer's-like beta amyloid, even though there's no evidence that they have the genetics for it, nor that they have any sporadic mutations or anything else, suggesting very, very strongly that indeed in humans that you can actually transmit an aberrantly folded protein from one individual to another. So this actually takes Stan Prusner's paper to its next logical conclusion, and that's probably the reason why he rushed really quickly to get those back-to-back papers out in the middle of July before this paper came out, because he wanted to have the first credit that you could actually show that in human samples that you could actually transmit a neurodegenerative prion-like protein. But this is actually the better paper. If you have a chance, go check it out, read it. It's fascinating reading. He's a great writer, and we'll come back and revisit elements of that at some future point. I'm just going to finish off with these last series of slides. So, some of you might be taking courses related to sleep. Have any of you taken any CSB courses related to sleep? So you know all about sleep, right? How does sleep work? It's complicated. Okay. It is. It is actually really complicated. Or maybe it's not. So I'm going to try to convince you that maybe it's not so complicated. This paper came out yesterday in Genetics, one of the premier journals in genetics. And one of the things that was really interesting about this paper is that even though we've been looking at sleep for over 150 years from a neurobiological and genetic perspective, and we know that sleep is actually really, really important for us. So it's been implicated in a lot of things like recharging, metabolic resetting, allowing your memory to consolidate during sleep, etc. We know that sleep is really important, allowing your lymphatic or glymphatic system to remove toxins while you're sleeping, etc. And we know that if you don't have sleep, that it directly impacts your health. And one of the things that you can see here is that there are very, very few neurons that you can actually see within the body of C. elegans, which also sleeps, by the way, and it sleeps a lot like us, right? So when you're sleeping, you kind of let your muscles go and you kind of lie down. The C. elegans does something very, very similar. So sleep is something that is well conserved. And C. elegans, as some of you will know, I really like C. elegans because the genetics are all known about C. elegans. We know every single cell in C. elegans, 302 neurons. We know every last cell within C. elegans. We can fate map every last cell within C. elegans. We know most of the neuronal connections within C. elegans, and we know their behaviors, and we know their genetics. And so there's lots of reasons why we really like C. elegans. C. elegans undergoes a particular type of behavior known as lethargus. And this is sort of a rest period that they undergo as they go from one developmental stage and they molt their cuticle as they grow bigger and they go from one larval stage to another. They kind of look like they're hibernating or sleeping, and this is known as lethargus or lethargus quiescence. So it looks a lot like sleep while they're doing this molting. So they have inactivity. They don't move around at all. They kind of lie still. They stretch out their body. They're not curled up. They stop feeding. They don't have pharyngeal pumping activity. That specific posture, as I mentioned, C. elegans likes to be in this sinusoidal shape. But while they're in this lethargus quiescence, they're linear. They look like they're lying down. And overall, they have a reduced response to reverse aversive stimuli. So if you try tapping their head, they don't respond right away. Just like if someone's trying to shake you up from sleep, that you probably also aren't going to respond right away as well. We know that you can get them out of this lethargus quiescent state with enough stimuli, and that if you do, it's rapidly reversible. Just like once you're awake, you're fully awake. And in C. elegans, we know that this quiescence is under homeostatic regulation. We've known for a while, and this is one of those big things. I kind of joke around about this a lot with students, but you can actually kill yourself by not sleeping. It's been shown in animal models from the early 2000s, and actually before that, Kushida showed that in 1989, that if you deprive animals like mice or rats of sleep, they'll die. We know that you can do the same thing in C. elegans if you continually tap them so that they don't undergo this lethargus quiescence, that they also die as well. Like sleep is actually very, very beneficial for you. We take sleep for granted, and we know that overall sleep has a number of functions that go beyond the scope of this course. And again, if you have a chance to take a fourth-year physiology or third-year or fourth-year CSP course, I really would suggest that you do so. But how about you? Do you think you can go without sleep? And if you can go without sleep, because I know some of you have, maybe you've done that yesterday night, for example. Maybe you'll do that tonight. How long can you go without sleep? 1 day, 2 days? Anyone stay up longer than 2 days? Ooh, okay. Anyone longer than 3 days? Ooh, okay. 4 days? Sorry? Not quite yet. Well, we know that overall, if you can't sleep, and this is sort of why I'm talking about prions. So prions are, yeah, really, really cool. And prions, you can get them from eating contaminated food sources, or you can have them coming in from a variety of other sources that misfold the normal endogenous prions. But what if you have one of those conditions that we talked about, either GSS or in terms of FFI? We know that this sleep is actually very, very beneficial for a variety of reasons, and we know that there's a lot of regulation of sleep, okay? One of the things that I highlighted here for you, and this is sort of the next topic that we're going to look at for neurogenomics, is that a lot of molecules have been implicated to be involved in sleep or regulating sleep. Some of these, whether you're looking at C. elegans or you're looking in the human brain, involve neuropeptides. And we're going to talk about some of those neuropeptides in the context of prions in the next lecture. In this particular paper that was published in Genetics, by the way, and I would like you to know this fact, in C. elegans, one of the factors that really regulates sleep is actually the epidermal growth factor receptor. And if you have the epidermal growth factor receptor, it's enough to cause you to go to sleep if you are expressing epidermal growth factor receptor in what is known as the ALA interneuron. And there aren't very many ALA interneurons in the C. elegans. In fact, there are only 2. And so only these 2 neurons are enough to control all of the sleep patterns that are found within C. elegans. So think a little bit about this. Is it any different than your hypothalamus? Or is it any different than how you sleep? Are there small groups of neurons that actually control your sleep levels? And are they actually run by things like neuropeptides? And what happens when you don't sleep? So here's where I need some of your help from a genetic standpoint. So what can you tell me about what you're seeing here? Genetics. It's pure genetics. Anything jump out at you? So we're looking at 5 different generations of individuals that might be carriers for FFI. What else do you know about FFI? Yes, or fatal familial insomnia. Yes. Yep, it is definitely dominant. It's found in every generation that we take a look at, and it is usually lethal. And unfortunately, or fortunately, as far as we know, when we take a look, there are only 50 families that have this unique mutation, where there is a mutation in the prion protein that causes them, after the age of about 40, to never be able to sleep again. So you can take as many tranquilizers as you want. You can engage in mindfulness meditation if you wish, but you will never sleep again. And after about a month, you die. And that's about the length that we know that a human can survive without being able to sleep. And this is a very, very interesting, but also really scary condition because it also resembles other things. So again, you're neurogenomics students. What other conditions that you know are related to sleep? Like either sleeping inappropriately or sleeping too much. What other sleep conditions do you know? Yep. Snoring, not really sleep, but okay. Yes, Kai.
Is it constant fatigue syndrome?
Chronic fatigue?
Yeah, chronic fatigue.
Nope, not chronic fatigue. Anything else that you guys are aware of? Yep. Narcolepsy. So we'll talk about cataplexy, narcolepsy, this whole idea of how things relate to prion disorders and fatal familial insomnia in the next lecture. And this goes back. Are we really all that different then C. elegans. Are there only a small group of neurons that actually control our wide behavior like sleeping? And I'm going to make the case that maybe this is actually a really good model for us to take a look at, and maybe prions and the damage that occurs after prions start to accumulate in one area of the brain is enough for us to lose our ability to sleep. Do you guys have any questions about any of the lecture material or anything else? Thursday will actually be a sort of a mini workshop on how to do well on your presentations, a little bit more about your grant. I just had permission from a student who wrote the grant last year to use his grant as an exemplar, so I'll be posting that up for you as well, and I'll also be posting, or someone in the office will be posting your group marks for all of you, and you'll get your adjusted score, and I'll also be uploading the midterm. Please don't go and upload it elsewhere, but I want you to see what the answers are before you get your midterm back on Thursday, or your group midterm on Thursday. You can come in individually to see me later on. Any questions? No? Any of you have midterms tomorrow? Some? Okay. Well, good luck on your midterms. Try to get some sleep if you can. I'll see you all on Thursday.